Welcome to Zoology Wala and myself Dr. Sai. Today we are going to see blood groups in the human population. Blood groups was initially observed by the scientist with the name Carl Landsteiner, and he discovered different types of blood groups in the year 1930 itself. And Bernstein, he identified that a gene is responsible for different types of blood groups, and the name of the gene was given as I, capital I, and is located on chromosome number 9. Generally, gene has two alternative forms. Either it exists in one form or other form. For example, if you observe the length of plant may be tall or short, like that. And even position of the flower, it may be axial or terminal, like that. But here, the gene exists in more than two forms. And anyhow, those alternative forms should be called as alleles. But here, the gene exists in more than one form. If the gene exists in more than one form, we have to use the word multiple alleles, and the concept should be called as multiple allelism. And we will see what are those different types of alternative forms. Generally, in population, the blood group will be given as A, B, AB, and O. These are four different types of most common blood grouping system in ABO blood grouping system. And the name A was given because of the antigen which is present on the surface of RBC. Antigen is nothing but it's a carbohydrate moiety which is attached to lipid and which is projected on the surface of RBC. In a simple terms, it's a different structure present on the RBC. And later they observed that another different structure is present on the surface of RBCs of some different persons. And one of the RBC, on the surface of RBC, they identified a different structure and they have given the name as capital A. In other organisms in the human population, they identified a different structure other than this B. So now, as already A name was given to that specific structure which is present on the RBC, and now they have given the name as B. And over the surface of RBC, in some people in the population, they identified the structure of A and the structure of B. Both these structures were identified on the surface of RBC. So both A and B antigens are present on the RBCs of some persons and on the RBCs of other persons. There is no question of any other antigen identified on the surface of RBC. As there is no antigen present on the surface of this particular person, they have been given name as O, 0. No antigens are present on the surface. And what about the antibodies which are present? What about the antibodies which are present in these blood group having persons? Generally, antibodies are present in the plasma. What type of antibodies are present? Here, what type of antigen is present? A is present. That's the reason why it is A blood group. What type of antigen is not present? And here, B antigen is not present. So, the B antigen is missing here. For that missing antigen, antibody will be present. It means in the persons of A blood group, B antibody is present. Similarly, in the persons of B blood group, A antibody is present. You can write it as an anti-B, once again, anti-A. And here in the AB blood group, there is no question of any antibodies. Why? Because both the antigens are present. And here, no antigens are present. So, obviously here, both anti-A and anti-B, both the antibodies are present in those persons. Both the antibodies are present in those persons. So now, what type of antibodies? Both the type I will write here, anti-A and anti-B. Both the type of antibodies are present in the persons having O blood group. So these are the antigens and antibodies present in blood groups of different types of persons in the population. And now what happens if you or if at all there is any mixing of different types of blood groups? For example, imagine the person is having A blood group. What happens if B blood group mixes, or if B blood group is given to that person? And similarly, if the person is having B blood group, what happens if you give A blood group to that person? That we will see now. And one more thing here, how we are going to identify whether the person is having A blood group or B blood group? How you are going to identify it? Simply, in a simple terms, I will explain. For example, if you want to know what type of blood is present in the given person, let us take one glass slide and take 2 drops of blood from the person. Okay, for example, imagine we have taken the blood from the patient. We don't know what type of blood it is. Take 2 drops of blood from the same patient and add the serum. Serum is nothing but the liquid which contains antibodies, obviously. So serum containing anti-A is added to 1 drop. Serum containing anti-B is added to another drop. And now what happens? Imagine if anti-A. What about the action of this anti-A? Anti-A works against antigen A. For example, if at all A antigen is present here, then there will be a clumping of blood. Clumping is nothing but breaking of blood. You can use the word agglutination also. It means here A antigen is present. If A antigen is present, then after adding anti-A antibodies will kill A antigen. Imagine after adding anti-B there is no reaction. Why there is no reaction? Antibody B is not going to kill any of the substance present here. It means anti-B can kill only B antigen. It means here B antigen is not there. That's why the reaction of anti-B is not here. It means if clump forms only with anti-A, this blood contains only A antigen but not B antigen. A antigen is present in A blood group. So the given blood belongs to A blood group. Similarly, once again, I have taken another slide. I have taken blood from the patient, from a different patient. I have taken 2 drops of the blood once again. To 1 drop of the blood, as usual, I have added the anti-A imagine, and to this I have added the anti-B. Imagine A antigen is not there. So after adding anti-A, there is no clumping. Why there is no clumping? Why? Because A antigen is not there. Anti-A will work against only A antigen. So that is the reason why after adding of blood, there is no reaction here. It means there is no antigen. Imagine here B antigen is present, and after adding the anti-B, what happens? Here there will be a clumping of blood. It means clump forms only with anti-B but not with anti-A. It means here only B antigen is present. Then we can say the person's blood group is B blood group. So if clump forms with anti-A only, that is A blood group. If clump forms only with anti-B, that is B blood group. And now see what I am going to write now. I have taken once again another slide. Taken 2 drops of the blood from the 3rd patient, and here I have added the anti-A, here I have added anti-B. After adding anti-A, blood underwent clumping. After adding anti-B, blood underwent clumping. So why the blood underwent clumping here? Why? Because A antigen is present here. That's why opposite reaction is observed. Anti-B is added, then the blood will break. So here what type of antigens are present? Both A and B antigens are present. Both A and B antigens are present in AB blood group. So the person's blood is AB blood. And by that we can easily come to one conclusion. What is that conclusion? I have taken the 4th slide now. I have taken the blood from the 4th patient. I have taken 2 drops of the blood. Remember, we have to take two drops of the blood. And here I have added the anti-A. And to this drop I have added anti-B. After adding anti-A, no reaction. Why? Because there is no A antigen here. After adding anti-B, there is no reaction here. Why? Because there is no antigen B. So as there is no antigen B, antibody B, it can't kill anyone. And similarly anti-A, no reaction after adding anti-A. Why? Because here A antigen is not there. A antigen is not there, B antigen is not there, no antigens are present on the surface of RBC with reference to A and B. That's the reason now, as there is no question of having any antigens with reference to ABO, the given blood group is O blood group. O blood group. So like that, if a serum after adding the serum of anti-A, if clumping forms, that is A blood group. Serum containing anti-B, if we added clumping forms, that is B blood group. If the clump forms after adding both anti-A and anti-B, then that is AB blood group. If no clump forms after adding anti-A or anti-B, or anti-A and anti-B you can call. So to 1 drop I have added anti-A, to another drop I have added anti-B. No clump forms with both anti-A and anti-B sera, then we can conclude that no question of any antigens with reference to ABC, that is O blood group system. So that is O blood group. Like that, in the population, see, we can't identify all the blood groups in the same person, isn't it? So in the population, we can identify whether the person is having A blood group or B blood group or AB blood group or O blood group. When a very simple examination in the lab, we can perform this identification technique. With that, we can conclude whether the person is having a or B or something like that. Am I clear? But here the point you have to remember, O blood group is not having any antigen. That is the reason why O blood group can be given to any of the other blood grouping persons in ABO blood grouping system. Why? Because there is no antigens on the surface of RBC. What happens? If no antigens are present, no reaction will be observed. Okay. Imagine A blood group person was given with O blood group. What happens? No antigens are present on the O blood group. So A blood group, even though it contains anti-B, it can't kill any antigen. Why? Because O blood group is not having any antigen. As O blood group is not having any antigen, O blood group can be given to any other person. That is the reason why O blood group is called universal donor, and similarly AB blood group is called universal recipient. He can receive blood, he can accept the blood from any person. Why? Because AB blood group contains no antibodies. Nobody is there to harm in AB blood group. So AB blood group contains, see, this is anti-A, this is anti-B. No anti-A, anti-Bs are here in case of AB blood group. So they can't kill any of A antigen or B antigen. So if A blood group is taken, they will accept. If B blood group is given, they will accept. If O blood group is given, they will accept. Why? Because AB blood group contains no antibodies. So that's the reason why AB blood group is called universal recipient. They can receive blood from any persons. But so many things are here, so many interlinking aspects are here. Even though O blood group is called universal donor, simply we can't give the O blood group to any of the blood groups very easily. So many technical issues are there, but this is just for preliminary information. That's all about different types of blood groups. Thank you.